Our speaker today is Dr. Carrie Adams, who's an associate professor in the U.S. IFAS Environmental Horticulture Department. Dr. Adams researches restoration of degraded ecosystems, particularly the transition from invasive species dominance to native species establishment. The collaboration with ecologists and other disciplines in cooperation with land managers, her work focuses on scaling up natural experimental manipulations to become application in natural resource management. And she's going to talk about salt marshes and living shorelines today. So thank you for being with us, Carrie, and I will turn it over to you.
Thanks so much. Thanks so much for the opportunity to speak today, too. I'm really glad that Laura and Savannah asked me to talk on this topic because a lot of my work has focused on Spartina for the past couple years. But this is really my first opportunity to talk about it all together. It's my first time, so thanks for joining me. It's been a really exciting research focus. We've learned a lot, but we've raised a lot of questions too. And that's why this talk is called What We've Learned and What We Wonder. So we're going to be covering both categories today. And the emphasis is really on we, because I'll be reporting on work that was done by my team of graduate students and my collaborators. So I'd like to just start by acknowledging some of those people. Laura and Savannah have worked with me on this, as well as Xiao Yu from engineering and Christine Rohal, biologist extraordinaire. And Vincent, Carolyn, and Holly, Florida Sea Grant agents, have been really supportive of this work and generous with their insights. Annie Roddenberry and Eric Latimer have also really contributed to keeping the work relevant and interesting to in a practical sense. And then from the native plant nursery side, Gil Sherrill, Joe Hayden, and Zach Shang have been generous with sharing their knowledge. Graduate students Madeline, Stasha, and Tori have done the bulk of this work, and I'm still learning from them. And our undergrads have been a great group of folks. They counted a lot of seeds. They've counted a lot of blades of grass, and they've gotten up at 4 a.m. to do field work timed with the tides. So I really cannot say enough about that group of people. We would not be where we are without them. So yeah, let's get started talking about Spartina. First, I'm going to start with a little bit of background on ecological restoration goals and how that links to our topic today. And then I'm going to tell you about 4 separate research projects. So this is like a 4-for-1. We're optimizing your value for the webinar today. So ecological restoration goals, and I just want to set the context for the whole work in general before we get down to talking about the Nature Coast. You know, the founding principles of ecological restoration had set as goals kind of taking degraded ecosystems and returning them to some kind of perceived historical state that you could picture, something pristine that left humans completely out of the picture. Since then, we are now in the UN Decade on Ecosystem Restoration, and goals have become a lot more evolved. They involve a recognition that ecosystems are dynamic and the environment is shifting, and it involves setting goals that anticipate that kind of change and future conditions. So really forward-looking kind of look at setting restoration goals. And then also, interestingly, goals increasingly incorporate social values, human interests, and stakeholders, and they're usually much more likely to reach their goals if, in fact, they are including humans and stakeholders. So I'd like to kind of track that shift in goals because it's relevant to our discussion today. Plants are really key to many of the ecosystem functions that are linked to these restoration goals. So let's talk about where plants come into the picture here. When you're revegetating an ecosystem, which is often a major component of restorations, you need to select sources that contribute to your restoration goals. And there's a couple of approaches that are kind of typical and commonplace and well supported by the literature. First, there's local adaptation, and this is when we take plants that are generated from populations that are adapted to specific environmental conditions that are similar to our restoration site. So we're grabbing plants that are locally adapted and introducing them to our restoration site. And we do this because it provides an ecosystem compatibility that leads to improved survival. So you're going to be more effective if you're choosing locally adapted plants. And then a less well-tested approach, but one that certainly is supported by the research, is the diversity approach. And what that entails is plants are sourced for your restoration, including multiple species, or in the case of monotypic systems, multiple genotypes within that one species. So choosing a diversity of genotypes or species leads to enhanced resilience and adaptability to change. So that's really relevant for dynamic coastlines like the nature coast. So these goals are really linked to important ecosystem functions, and they have unique relevance in coastal systems. And that's because of the outsized contribution to ecosystem function that coasts have. So by restoring them effectively, we're getting more bang for our restoration buck. They are uniquely high value, biodiversity hotspots. They support dense human populations. All this is not news to this group, but also we know that they're vulnerable to rapid degradation. So it sort of lends increased urgency to our restoration efforts. A lot of these coastal environments include plant communities that are largely dominated by a single species, like mangroves and salt marshes. So that brings us to our, our species of interest today. Spartina alterniflora, this is smooth cordgrass. It's a keystone species that ranges from Maine to Florida to Texas, so it's got a wide ecological amplitude. It also has morphology that features these dense below ground stems as well as this above ground architecture. And these two things together make it an ecosystem engineer. So it modifies its environment. In this case, oftentimes providing shoreline stability. So let's take a closer look at what that morphology entails. We got the dense root and rhizome network, enhancing soil structure and promoting carbon storage. And so we're really supporting nutrient cycling. It has a really high photosynthetic capacity for a couple reasons here. And that means it has enhanced primary productivity and really gets that organic matter into the sediment. So important to coastal systems in that way. Now, here's something else to think about with Spartina. It reproduces asexually via tillers and rhizomes, so kind of spreading vegetatively underground, but also sexually by seeds. And so this bimodal means of reproduction means that the species can be pretty resilient in dynamic coastal environments. So it's all of these characteristics that make Spartina a great restoration species. So it's planted for restoration and enhancement, to replace eroded marshes, to trap sediments and provide coastal habitat. And then a special kind of restoration or enhancement, I guess we'd call it, are living shorelines. And I'm sure that living shorelines are commonplace to a lot of this group, but living shorelines utilize vegetation and natural elements to protect the coastline. And they offer an alternative to hardened structures, and they're increasing in their implementation and really garnering interest at a national level as well as a state level, and interest as well along the nature coast. So that is why we are interested in planting Spartina for restoration. Something else that we need to know about Spartina is that research on populations of Spartina that have been done outside of Florida include they come to the conclusion that these populations have population-specific traits, and those traits persist when the plants are planted elsewhere. This is all consistent with local adaptation. So we're assuming that Spartina alterniflora has this degree of local adaptation, but it's not very well researched in peninsular Florida, which is where a lot of the restoration and living shoreline action is. in Florida. So genetically diverse Spartina also, we know from these other studies, is more productive and provides more function. But this is definitely context dependent, has to do with stress. It may be climate, maybe might alter the situation. So we need to know more about Florida Spartina, basically. And that brings us to the 4 studies I'm going to talk about today. So projects 1 and 2 are one is wrapped up, 2 is wrapping up, and then 3 and 4 are newer studies. So, but they all kind of fit together in an interesting way, as it turns out. So we'll start with project 1, how to best plant Spartina for sediment stability. That was really the main question that we had when we started looking at Spartina. You know, if it's going into all of these projects, are we sourcing and planting in the way that's going to most best stabilize sediment? So what we did was we started looking at the traits of several different populations in Florida, and we included not just living shorelines, but also nursery sources and donor marshes. One of the things that we noticed when we started looking at these sites was also not only did the plants look pretty different, even though they were the same species, but also some of these were growing in sandy environments and some were growing in organic matter environments, and it looked pretty different. The soil did, and we'll come back to that distinction later. We also know that the industry standard is really to plant Spartina in rows with 1 plant every 1 to 3 feet. But there's really not a lot of information on testing that approach to see if that's the best for sediment retention. So we did some experiments. We started off with a common garden experiment in which we took individuals from all these populations and planted them in exactly the same conditions to see if their traits, differences in their traits were maintained. We also did a mesocosm experiment, and we wanted to compare a few different sources of Spartina in contrasting sediment conditions under different planting designs. So manipulating those things to see if we could figure out which would be the best approach. We took all of those measurements that we did on plants on Spartina. It was a lot of measurements, and we used that to better parameterize a numerical model. This model was developed by Xiao Yu, and it began with kind of a typical sediment dynamics model in which we have just kind of dowels stuck in sand. And one of the benefits of our experiments that we were able to better parameterize this model to give more realistic parameters in terms of what the plants were representing. So the common garden experiment, we'll go over those results first. Yeah, it turned out that the plants acquired from the 6 different sources really express a diversity of traits. We're looking here at number of shoots, clump diameter, and mean height from each of our accessions, and we see a diversity of traits, and also notice that there's no source that kind of is best for sediment stability. Depending on what we measured, each sort, there was a different winner each time. So that was interesting to us. We also did some genetic analysis and discovered that genetically similar sources had similar traits. And so that's kind of the first clue that we had that it might not just be environment alone that's shaping these plants, but genetics as well. Okay, the other experiment we did was this planting design experiment in cattle tanks. And we looked at these 4 planting designs, which you can see the green dots in the top of this diagram. And we planted 2 different sources, and we planted them in organic and sandy soil. What we found out was that the typical planting, just this 1-foot centers, that was outperformed by double, triple, and clumped planting designs. So any of the alternative planting designs outperformed typical density in 1 to 4 months. We also found that each of the sources had slightly different sediment trapping advantages, and that they grew slightly differently in the two different sediment types. So we're starting to see that the source matters, the environment matters, and the planting design matters. Um, so we took all of that information and plugged it into this priorly oversimplistic sediment transport model. And we published this stuff just this year, and I'm happy to share the citation for anybody who's interested. Um, but basically, we utilize those measurements from our experiments in the model. And I'm going to talk you through this. This is our model results. Red means erosion, and blue means sediment retention. And we've got four different scenarios here. You can see that we looked at Spartina arranged in rows, and we also looked at large clump sizes versus small clump sizes. And we just look for the simulation that has the most blue in it, and that's this one. That is larger clumps planted in random arrangements. So we thought this is a pretty interesting outcome from the model. We haven't found a way to test it in practice yet, but the model is suggesting that these random arrangements of larger clumps is a better way to go out trapping sediment. We also, through other model runs, looked at other issues. We noted that below ground biomass offered by Spartina provides the netting effect to protect sediment from entrainment. So that lower below ground biomass is pretty important. And that's, by the way, one of those things that differed with each source. Also, we discovered that organic sediment was actually more likely to resuspend and be less stable. And so there's a different challenge to be overcome regarding sediment retention based on sediment type. So that's project one. From this, we learned that the potentially a best planting design that we'd like to test in the field. We learned that sources had different traits. We learned that population genetics may play a role. But we were also that one of our main findings was that source really matters for selecting plants for your restoration. That brings us to our second question. Anecdotally, we had heard of issues with Spartina supply limiting project completion for these restorations. Now, there isn't a lot of widely available information on plant material sourcing really for any species, and Spartina in Florida is no exception. So, we just don't know a lot about this, but we had heard both from vendors and practitioners that lack of Spartina was limiting projects. So we performed a Qualtrics survey of Florida practitioners who routinely plant Spartina. Our contacts were from the Florida Sea Grant Marine Contractors course, as well as the Florida Regional Estuarine Restoration teams, and we used a snowballing approach. So we asked them to add anybody that they knew who was planting Spartina. One of the most insightful questions we asked was, what challenges have you faced when sourcing Spartina? It was interesting to see their challenges. Budgetary constraints we usually think of as being whatever, the main limitation to restoration. But there were some other answers here. In fact, the top three choices had to do with availability. Limited choice and source was the top answer. So people are just not able to get the source. We've got supply is an issue, but also specific sources is an issue. So looking a little bit more closely at these data, we then asked if challenges in sourcing Spartina were encountered, how did your plans for this project change? And this was pretty revealing. We saw that almost universally the answers were pretty negative, kind of cringy. They had to harvest or propagate plants themselves, which is a massive increase in labor and time. Use some kind of less desirable plant at a lower density, plant a smaller section, just things that it's a shame that these were kind of what was coming out of just simply not having enough plant material. So what we got to thinking about is that, we take these two things together, and it definitely seems like, given these challenges, improving source availability could improve the capacity of Spartina plantings hard stop. And we got to thinking, vegetative propagation is pretty labor intensive and it could lead to reduced genetic diversity. What else could we do? A lot of other restoration species are seeded. So we started looking at seed. Seed might provide a source with less investment required. It definitely gives you a lot of control over source. You just collect the seed from the source. And so now you're asking, why weren't people doing that already? Well, anecdotally, we had heard from practitioners and vendors that they were interested in seed, but they did not have a lot of success with it, usually because germination was limited or unpredictable. And actually, we don't know a lot about Spartina germination from the South. We know a bunch from Heather Joesting's lab at Georgia Southern, but as far as Florida goes, there's really limited information. So that brings us to actually researching this question. Can we improve Spartina plant material supply with seed? This work is from Madeline Estes, who defended earlier this month, and she's in the middle of preparing her publications. And she went out to 10 different marshes and just collected seed. One of the things that we didn't realize was going to be such a challenge was timing that collection to be optimally ripe. It turns out that these visual cues are really important, and timing makes a big difference in whether the seed is going to be ripe enough to collect. And so that was kind of step one of this work, was trying to figure out when to collect seed. And of course, because Spartina is not the most cooperative species logistically, it's a peak shopping time. So between Black Friday and Christmas Eve seems to be the best time to collect this species in Florida. We still have a lot of questions about that too, though. So Madeline did manage to get out and collect Spartina, and we started looking at these 10 different accessions she collected, and it was just really interesting to see. The first thing we did is we looked at how filled this, we looked at the seeds carefully on a light table and found out that a lot of them did not have embryos. Some of them did, and some of them didn't. And we confirmed this with the X-ray. And now I'd like to show you the data. Look at that. Percent seed fill tops out at 30%. Pretty interesting. And look at the difference between all of these populations is definitely statistically significant. We found that the seed fill differed with the source significantly. Also interesting, restored marshes produced the most filled seed. So we started to notice these differences based on restoration history. Another really interesting finding, going back to that time of collection issue, we had 3 accessions that we were able to collect 2 times. They were only collected within 2 weeks of each other. And look at that difference in fill. 1/3 more fill collected at an earlier time for CKA, for instance. So collection timing is definitely a question that we have. Okay, so that's fill. We looked at fill. And then from the filled seeds, we looked at germination. So when the embryo actually emerged from the seed coat. We did these tests on filled seed only. And what we found was that germination percentages also varied with source. You can see that here we've got significant differences. Another thing that we learned was about cold stratification. Seeds in the Northeast often have a physiological dormancy that if they're kept in cold storage for a while, germination will then be induced. We were not sure whether or not we would see that kind of dormancy in Florida Spartina. It turns out we have pretty good evidence now to suggest that stratification revealed this kind of dormancy. Check it out. The 3-month stratification is, we got more germination from every seed lot. with increased stratification, to the point where the MDC accession, we got nearly 100% germination of filled seeds if we cold stratified them for 3 months. So this was a real breakthrough. Germination differed with source and it improved with stratification. We also noticed again, we only had a couple natural marshes, couple restored, and couple donor marshes. So this definitely put this under the category of what we might be wondering about. But natural marshes germinated less than restored and donor marshes. So another interesting finding that we wanted to follow up on. Okay, now we took the seeds that germinated from that experiment and we hung on to them. And then we took those seedlings and we planted them in the greenhouse to see how their traits persisted. Measured a lot of stuff, but I'm only going to focus on these. We had different accessions, had different clump diameters, different number of shoots, different number of leaves, and different survival and different biomass. So significant differences in all of these sources. PMK was the most productive, shoots, leaves, and biomass. CKG2 was the least likely to survive. And MDC had smallest plants and was the least likely to survive. Interestingly, if you remember from the last slide, PMK was the accession that had the lowest seed fill. So we had the lowest seed fill and the largest plants. MDC, alternatively, had really high seed fill and then plants that were kind of wimpy. So there may be some trade-offs going on here that we would like to investigate more. But it definitely suggests that trying to decide on the best accession based on plant traits for sediment retention is a complicated business. So the other thing that this got us thinking about was, so we know that source really influences performance. Mesocosm experiments showed us that environment matters too. We think that Spartina exhibits local adaptation. What is going on in natural Florida populations? Do we see that kind of evidence for local adaptation? That brings us to our next experiment. The question is, how do Florida Spartina population traits differ with source? And this is Tori's dissertation work. Tori went out to 6 natural populations that were kind of regionally distributed in order to try and understand these regional differences in natural Spartina, to kind of think about how local adaptation is playing a role. Is it the case that the Nature Coast populations are different than the Atlantic Coast populations, for instance? So she measured in the field a lot of parameters and really got to know these plants well. I'll show you the results of what she measured and how it differed with each source. And again, we're seeing these significant differences in traits based on source. Also, we're seeing that the winner is not the same in every diagram. So we're seeing consistency in the story. But that got Tori thinking, what could be the explanation for these differences that we're seeing? It could be genetics. It could be the age of the site. It could be regional characteristics, disturbance history, other things too, probably. But we're starting to reveal these differences that make us really curious about, why do these differences exist? Tori set to answer at least a little bit of the question with a common garden experiment. And the question she was asking is, do these morphological distinctions that she measured in the field, if we plant those plants all in the same condition, do those traits persist? So she took 10 plants from each source and she planted them in the greenhouse and let them grow. The measurements showed that, in fact, we did see a little bit of shuffling when the plants were grown in the same environment. Some of the traits were retained over time. Some of the smallest plants tended to stay the smallest, but others, there was a reshuffling of the largest and the smallest based on measurements of their sediment retention related traits. So from Torrey's Common Garden experiment, we know with the natural populations, source matters, and then also environment matters. So it depends on where the plants came from and where they're planted. So it makes you wonder again about the genetics of the species of our collections that we've found so far. And you wonder how genetic identity within and among populations might be related to those trait expressions in the field and greenhouse conditions. And then what would be the implications for restoration for these species? What does it mean for these accessions? What does it mean for how these plants provide ecosystem functions? One mechanism, and we're going back to those special traits about Spartina, one mechanism that underlies these distributions that we see in genetics and traits is seed dynamics. And that may be the thing that we know the least about for Spartina in Florida and elsewhere, actually. And that leads us to Stasha's master's work. Her question is, how does Spartina spread by seed in nature? And certainly, we just don't know a lot about the relative contribution of vegetative spread versus seed dispersal and germination. The whole process of Spartina regenerating by seed we'll refer to as seed dynamics. So it's like how the seeds interact with their environment to become real plants. Stasha made this nice schematic. We look at how seed arrival at the site and then seed retention at the site leads to what we can call seed availability. Is the seed even there in the first place? Then the seed might germinate and turn into a seedling and survive into an adult. So that's kind of the path of how seeds contribute to the extant plant community of Spartina. Seed dynamics can be affected by a lot of environmental variables. We did not measure every single one of these, but Stasha is looking at how vegetation and vegetation density might be influencing seed dynamics. She designed two really clever experiments to try and answer this question. The first thing that she did was she's in the process of looking at Spartina marsh sites to determine what seed arrival and retention looks like in the field. It's a challenging field experiment, but it's so far so good. Stasha also investigated the impact of seed availability and extant stem density on germination and seedling traits through a greenhouse experiment. Let's take a look first at her seed arrival and retention project. This is Stasha in the field, and she has two types of traps, an aerial trap and a ground trap. And she's kind of trying to get at all the potential means of dispersal by collecting seed with both of these traps. She also noticed that there were a lot of bare patches within the stands of Spartina and kind of thought about, maybe I wonder if that might be an opportunity for retention or germination or survival in comparison to primarily vegetated patches. And her preliminary pilot data showed that she did see a difference in seed count between two different sites, but that difference was not as marked as the difference in cleared versus vegetated patches. So her theory is maybe testing out. Of course, we have really low numbers of seed. We set out many more traps this year at many more sites, and we're going to collect over time so we can hopefully catch that peak seed output for Spartina. But it'll be interesting to see what happens as far as seed arrival and retention in both vegetated and unvegetated patches. It's especially interesting given the results from Stasha's greenhouse experiment. She did this experiment looking at seed dynamics and the impact of existing vegetation on dynamics, and she did this in the greenhouse. She had two treatments that she varied. One is seed density. You can see these Spartina seeds. It was either 3 or 8 seeds. And then she had differing levels of extant adult plants, high, low, none, low, and high. And so looking at her results, first we'll look at germination. We were all kind of surprised to see that there was very little difference in germination between the treatments. It ranged from 33% to 58%, so it was a little bit different, but it wasn't statistically significant. And it was kind of surprising to see that there wasn't much of an influence in this when we measure germination. But wait, there's more. Seedling survival, on the other hand, did significantly differ with her treatments, and substantially so. And check this out: when your seedlings were 90 and 81% survival, I mean that's a really high survival, and that was in the clear patch. So it's suggesting that competition with above ground plants may be limiting survival. And it may be that the clear patches are related to how seeds make their way into the population and the genetics of these stands. How are these seeds contributing to this like bigger genetic identity, diversity, and trait expression questions? That still stays in the what we wonder column. But definitely interesting results, and I can't wait to see what she finds out this field season. So speaking of what we are wondering, I'm going to summarize everything for you that I talked about so far today. We'll start with what we learned. And when I say what we learned, some of this is still preliminary. Most of it is yet to be published, and there's other caveats that we need to be cognizant of. Like, we didn't have a huge number of sites. We only looked at 1 year. We had a limited number of populations. We're confining results to our study region. So, I got to get all that stuff out of the way, and just acknowledge that up front. But given all that stuff, here's what we've learned. Sources of Spartina matter. They differ, they got different traits, and they deliver different ecosystem functions depending on the source. So sources differ and sources matter. It's a challenge to acquire specific sources, which is kind of limiting our ability for these projects to provide their optimum capacity. Sources respond differently with the environment, so it's not just the source that's important, it's where it's being planted as well. It looks like with a couple tweaks, we can improve source supply with seed. It is definitely an option. There are logistic barriers remain, but it seems like germination might not be one of them. Natural marshes seem to be behaving slightly differently than restored marshes. And then also seeds look like they contribute to population, and where that is going to happen probably is in recruitment in these gaps. So that is basically, boy, that's a lot of experiments and a lot of work, but that's our list of what we've learned so far. And here's our list of what we wonder. I really trimmed this down. We wonder what the implications are of this source-specific performance for restoration. We know it's important. We know the differences are significant in our experiments, but what does it really mean on the ground? And what is that effect size, and how much investment is it worth to try and figure out what the right sources are and how much more the ecosystem function they deliver? That's a big question in practice and just in basic ecology as well. We also don't know a lot about how seed quality varies from year to year and within a year with a collection calendar day. So our evidence suggests that it could vary quite a bit. And so it makes you wonder, how much of the variation that you're seeing is just due to timing. That's a really big question still. We also want to know more about the genetic variation between the natural marshes on the Florida Peninsula. I'm really glad that Tori is starting to look into that, because it would be nice to know how much those populations differ from each other genetically, and if that's related to any spatial pattern in the landscape. And then the last thing we wonder is, what is the relative contribution of sexual and asexual reproduction to Spartina population structure? That has important implications for the functions delivered by any single population of Spartina and for how we should be sourcing the plants that we're planting in restorations. So that is what we learned and what we wonder. And I'm very happy to take any questions.
Thank you so much, Carrie. That was really great. And it was amazing to see all of those different projects come together in one cohesive talk. Thank you.
Thanks,
Um,
Laura.
if folks have questions, you're welcome to type them into the chat and I will read them out. You can also unmute yourself and ask Carrie a question directly. Um, I'll go ahead and start. I wanted to know if um, you talked a lot about using seeds and growing them up into adult plants to plant for restoration. Is there anyone who is putting seeds directly out onto marshes, and what do we need to know to be able to do that?
Boy, that is a great question, Laura. Deployment really seems to be the next step, doesn't it, in seed-based restoration. And in a dynamic coastal environment, we really have our work cut out for us. In areas where seed-based restoration with Spartina is happening, the only areas that I know of, and by the way, it's very hard to figure out exactly what is going on with seed-based restoration because a lot of people are doing it, sharing information with each other, and not necessarily publishing it because, they're too busy trying to get Spartina established. But from what I can gather, a lot of places that do direct seeding are in different hydrodynamic environments than we've been looking at. So it's kind of the back marsh, areas where there are substantial differentiation in elevation, creating potential pockets that allow the seed to be retained on site. But that is what I know about direct seeding. I am really curious too, and I feel like I would like to do another survey.
Great. I think Caitlin took herself off or put her camera on. Do you have a question?
Yeah, I have lots, but I will limit them. I also put a link in the chat. I run a program in Mississippi called the Native Plant Producer Network out of MSU's Coastal Research and Extension Center in Biloxi. We're kind of different than normal extension. We do, again, like very coastal based things. And this program is designed to get anyone and everyone growing native wetland plants for living shoreline projects. And the two that we're focusing on are Spartina and Juncus,
Mhm.
riverianus. So we do a lot of, like you mentioned, vegetative reproduction, as well as sexual. So we have permits and are collecting seeds across a lot of the coastal preserves in Mississippi, a lot of like private property, locations for Spartina. And I wanted to kind of plug the program in case there was any way we could potentially collaborate or do more. And I was curious about some of the genetic work because our group is actually traveling to Argentina in February, and we're doing a lot of salt marsh restoration type experiments. And the one that I'm kind of taking the helm on is I wanted to know what kind of differences genetically Spartina marshes in Argentina have depending on how close they are to population densities. And so I was curious, like, what were some of the genetic, like, markers or things you guys were looking for in some of these genetic studies?
Yeah. You should probably take me to Argentina with you. Caitlin, this is great. I just saved the chat so I can get to your website and keep your contact information.
Yeah,
I'm really excited about your suggestion and what you have going on there because one of the things that we've been struggling to find is, real live restoration that's happening with Spartina. So I'd love to talk to you
yeah,
more about your sites. That's
absolutely.
really cool. And we have found, I just looked at a little bit of Tori's data recently, and her differentiation indices just for populations that we found in Florida are like, they're pretty different. So it would be really cool to look at Argentina as well and add another like layer of scale to these genetics. That's really cool. Let's definitely keep in touch. Did I answer your question? I don't know.
Sort
I
of.
just kind of got excited.
I was just curious if there was specific genes or genetic markers that you
Oh,
were looking for in those, because I haven't spent a ton of time looking at
yeah.
it yet, but I know that potential stress genes are probably the ones that we're going to be kind of focusing on, obviously, if we're thinking about marshes that are next to large coastal cities.
That is so cool. No, we have not looked at stress genes. We've mostly been looking at diversity and similarity indices. But that would be a great next step.
Okay, cool. I have so many questions, but I'll wait until everybody else
Let's
can
connect
ask.
later.
Yeah.
Yeah.
There's a question in the chat from Emery, and she says it was a great talk. She asked if the regional differences you found, she says the regional differences you found were very interesting, and she's curious whether these sites differed in their wave energy exposure. She's not a geneticist, but wonders if wave energy tolerance may have genetic markers which could show up in seeds.
Dang. Hi, Emery. That is a really good question, and I think I better add that to my list of potential explanatory variables. Yes, wave energy. I mean, it would make sense that plants have all kinds of adaptations that are based on the disturbance regime in which they're growing. So it would make sense if they were adapted. And I think part of the impetus for our natural population study is to get as many populations as we can on the Atlantic coast and the nature coast because there's such a different wave climate. So, we haven't set anything up to which we can answer that question directly, but it would be interesting I mean, we'll be able to look at those differences regionally. Maybe we can infer and then write a proposal and then do another project to answer that question.
I see Heather turned her camera on. Do you have a question?
Yeah, hey, how are you?
Hey, Heather, good to see you.
Good to see you. It sounds like we're doing some pretty similar stuff.
Yeah.
Although not too similar. So I'm going to follow up on Emery's comment because it made me think, how did like the level of anthropogenic disturbance differ among your wetland sites?
Yeah, right. So I think the anthropogenic disturbance, I'm glad you brought that up. I think that might be one of the drivers that separates our natural restored and donor marsh sites. Our natural sites um, tended to be have a different suite of soil and abiotic variables than the living shoreline sites, and to the point where it was really hard to differentiate the differences that we were seeing between natural and restored sites to any single variable. And that I think has made us just want to get more sites to hopefully try and understand that question. But you're absolutely right that there's like an anthropogenic component to these plants.
Yeah, because I know one of the sites we go to is heavily boated.
Yeah.
And so I think about, kind of how the boat wake is
Yeah.
messing with my plants and messing with the reproduction. So
Yeah.
interesting question.
Yeah. And we know that stress influences seed production and seed quality and seed density and all kinds of other measures of fecundity. So it would make sense. Let's get a bunch of different sites that have different wave actions and measure seed production.
Does anyone else have any other questions?
I had one more in relation to seeds. Um, Whenever I believe you said her name was Madeline, she
Yeah.
was the one collecting a lot of the seeds. Do you know if she experienced seeing a lot of fungus growing off of them? Because I know in some of the marshes down here, I can't remember what the fungus is called, but it kind of looks like a little bear claw like coming out the top of the flower.
Yeah. Fungus, right? So I know that there are naturally occurring fungi that occur in marshes that seem to just leave Spartina alone. But I also know that, there's there's a variety of them, and every fungus has a different impact, right? Um, we did see a lot of fungus growing on our seeds when they were trying to germinate. And so, you know, part of me was concerned about, should we try and strip the seeds of any fungus that we, should we try and clean them really well? But there's so much about seeds that are related to symbiotic germination that I think we want to be really careful about completely isolating these seeds from their microbes if what we're trying to do is promote their health and germination. So gosh, Caitlin, I wish I had a better answer for you, but I think that looking at plant interactions with fungus and soil microbes and just kind of other I think a lot of the Spartina work has been about looking at consumers, but I think it might be time to look at different trophic levels too.
Gotcha. Thank you so much.
Thanks.
And there was a little bit of information in the chat that they're also seeing some of that fungus in Georgia.
Interesting.
Any one last question? We're getting to the end of the hour, and I just want to say thank you for the great conversation. Thank you for the great talk, Carrie. We will be back a little earlier than normal. Many of you might know our first webinar of the season was postponed, and it was postponed until November 5th, which is in a couple weeks. And that will be Dr. David Kaplan, who will talk about mangrove expansion on the Gulf Coast of Florida. So hope to see you back in a couple weeks, and thank you all for being with us.